And as always, after the Power Talk sessions, please scan the barcode that will be presented once it's concluded for your evaluation survey and your attendance. Okay, and without further ado, I give you Dr. Jian Ying Chuan as our first speaker for today to present. The floor is yours.
Okay, thank you. So today I would like to introduce how we can study nerve regeneration after traumatic brain injury, and what is the way we can treat for the patient or in some brain injury animal model. So next. This is the outline. I will introduce first for our cardiology and the neuroscience department, as well as I will introduce for the TBI and how we can treat the TBI. Okay, so next. First is Taipei Medical University. We just show you we have we established in 1960, and now we have 11 colleges. Also, we have 1,002 faculties, as well as with the staff members. And now we have 6,000 students. Okay, so next. Our college is one of the 11 colleges in Taipei Medical University. Our college name is College of Medical Science and Technology. Then we have 60 faculties already. Also, we have almost to 500 students. Okay, next. Here I just show you our campus located in the Shuanghe campus. This is our building. Next. And then we just have a 60 faculty, but the performance, we are best one of the colleges in Taipei Medical University is better than medical, College of Medicine. Okay, next. Now, why we have the outstanding performance, just because we have a wonderful leadership, also including me. But I would like to say our dean, she focused on the drug discovery as well as she has some industry background. So she lead us focus on the translational medicine, and then we can just apply our research to the clinical treatment. Okay. Also, we have two vaccine. One is Huang Pu. She is international vaccine to take care about the international issue in our college, as well as we have Professor Gao. She take care about the teaching. Okay, next. Also, we have a chair professor as a mentor and leadership guide our college to have several research team, as we have a diversity development in our college. Next. So this is our development map. We not only focus on the East Asia and East South Asia, but also have a cooperation in the Canada and the United States, as well as we have a cooperation with French and Australia. Next. So now in our college, we have 7 spring out companies, and then we are so happy that we have this spring out company, because that means our research is application in some area, especially for medical treatment. Next. Okay, this is our education structure. Actually, I can skip this slide. And now here we will share some research area in our college. Actually, we have 10 departments cover 5 major research areas. One is biotechnology. Next. Another is translational medicine, as well as the cancer biology, and bioinformatics. So all research field, we will invite our colleagues. They will introduce the cancer biology, translational medicine, bioinformatics, as well as biotechnology for you. Okay, and the next. This next. Okay, now I belong this program is a neuroscience program, and then I will introduce this program. Okay, next. Now in this program, we have 2 departments. One is a PhD program in the medical neuroscience. Another is the international master program in the medical science. In our program, we cover several areas of neuroscience, including neurodegenerative disease, traumatic brain injury, stroke, brain tumor, as well as for the development neuroscience. Next. Now we have 11 faculties already, as well as we collaborate with the National Health Research Institute in Taiwan. So this is the reason why we can cover so many research areas in the neuroscience, just because we have enough faculty resource to support the study. Okay, next. And then this is our equipment. Next. And this is our international students. Next. And I think this is quite important for everyone, just because, yeah, of course, we have the faculty to support the research, also guide our student to perform the research. But most important is, I just mentioned in our college, our faculty, they have a research grant, so they can support the student for the scholarship. As well as in TMU, we also set up the scholarship if you can apply the master degree in TMU or PhD degree in TMU. We just depend your performance. We will give you a type A or type B scholarship. As well as we work in our department or our laboratory, we will provide you add-on scholarship to make sure your life in Taiwan is no problem, okay? As well as, yeah, sure, for the program support for student, okay? Next. And if you're interested about the neuroscience, just contact, we have faculties show here, and then they are windows you can contact to get more information, or you can go to online to check our website. Okay, next. Okay, so this part I just would like to share my research. Do you know how we can build up our memory in our brain? Yeah, I think it's quite important. So this paper published in MIT, and then they just found when the mouse get a shock, some brain region like the basolateral amygdala will respond or receive the information from the shock. But if you combine with some environment synchrony, like a context, our hippocampus will treat this information. Then the hippocampus covered with basal amygdala, they can transfer the new signal to the prefrontal cortex. Although we create the memory circuit. So the mouse will remember when they stay in the environment signature. That means he probably will get a shock. So maybe next day when we just provide the environment signal, and then mouse has a behavior. But few days later, the mouse will forget everything. But if you repeat to provide this invalid signal combined with a shock, now in our brain, we will to digest this kind of signal, allow it become simple circuit. Then finally, you just use the prefrontal cortex and the basolateral amygdala. So we can quickly to get the response for the signal. So for example, how when you are baby, you learn how to use a chopstick. Then your parents will tell you you need to use a finger how to take the chopstick. Now, when you just take a chopstick, it's easy to pick up any food. Okay, you don't need to remember how we can take the chopstick. Just because our body and our brain will learn for every behavior or every body behavior. Okay, next. However, when we got the brain injury, for example, here we show the brain injury next. And we just got a primary injury next and the secondary injury. What is the primary injury and the secondary injury? For example, your skin got a knife damage from the knife. And then in the moment, you will feel ouch. But after half days or 1 day, you will feel so painful just because some inflammation in the skin. In our brain, similar, we got the primary injury just the beginning of the damage, but few days, our brain will extend the neuroinflammation, will extend the damage. So it will create some significant damage in our brain. Next. And then we can see this is the damage timeline. And the next. Then it will create some protein unfolding, allow the our brain has some neurodegeneration. Next. Then finally, we have some dementia or some brain disorder. Okay, next. So in our model, we just want to learn how we can study the traumatic brain injury. So we use the animal model. In our lab, we have two kinds of model. One is a weight drop, another is a control cortical impact. Next. So here we can just collect the mouse brain to stand neuroinflammation marker like GFAP. And then you can see, next. You can see in the brain tissue side, the damaged side is the right side. Sorry, in your is the left side. So you can see the red color is quite a significant indicator in our model. It's no problem for the brain trauma. And next, can you just push the movie? Touch the figure. Okay, so this is the brain 3D brain image, and then you can see the yellow color indicate is the GFAP, the neuroinflammation. The pink color indicate the neurodendrite. And then after damage side, you can see the pink color is disappear. And the yellow color is quite strong. So next. Next. So here just show you the image. In our brain, maybe you got the damage, but the fiber, the neural fiber, it got the damage is more severe than you see the tissue structure. So we want to know how we can let the neural fiber regrowth again or repair again after traumatic brain injury. Okay, next. So in our study, we focus on the sigma-1 receptor and the 179 because it's just a single pathway, so I don't want to spend too much time to introduce the detail. I just would like to let you know we found we can drive this single pathway allow some neuroprotective factor like antioxidant that highly expressed in the neural cell to protect the neuron against some oxidative stress following traumatic brain injury. Next. Or you can show all figure in this slide. And one more. Okay, so this is a sigma-1 receptor in my lab. My previous study, we found it have a several role in our neural cell. For example, like drug addiction and epigenetic regulation in our brain, as well as for the nuclear pore transport for the messenger RNA. And then it correlates to several neural disease. Next. So when I studied the sigma-1 receptor, is the one kind of ER chaperone protein, and next. And you can see it's a kind of receptor, so it can respond for the neurosteroid, allow it to be the active chaperone protein to change the target protein structure, allow it have a different function for the target protein. So next. So in here, we found the 179, DNF 179 is a downstream of the sigma-1 receptor. When we treat the ligand with the DHEA, it can let the 179 shuttle in from cytoplasm into the nucleus to trigger several gene expression. Next. And why we study the 179 just because it is highly enriched or highly expressed in our brain. And then it correlates to the neural development, but next. But we don't know what is the major function in the traumatic brain injury. And then we found the 179, this chaperone protein is able to enter with 179. So we just want to know whether this kind of a single pathway major function in the traumatic brain injury. So we perform the single cell sequencing, is a kind of anti-sequencing to study gene epigenetic regulation. Next. And then here, I think the figure is quite complicated. I just want to let you know, when we overexpress 179, one cell cluster around here, it create after the 179 overexpression, and then we check with several biological marker. We found is a kind of neural progenitor cell. Next. And then we also treat with the DHEA to make sure whether we get the similar result like a 179 overexpression. So here we use another sequencing marker, sequencing technique is spatial sequencing. Then we found the damage site after DHEA treatment. The 179 expression level is significantly higher, and then it creates one biological function is neural projection. Okay, next. Also, we check how many protein can enter with the 179 compare we treat with the cell with DHEA. Next and the next. And the next. Okay, so we found the 179 image locate in the cytoplasm, and when we treat the DHEA, we can increase the interacting protein with the 179, indicate the DHEA indeed it changed 179 function already. Next. And then we found major is a correlate to the cell movement and the biosynthesis. Next. Next and next. And one more. Okay. Oh, one more. Yeah. Okay. So I just skip some detail because the time is limited. I just would like to let you know. Finally, we check. Yeah, indeed, the 179 and the sigma-1 receptor, this single pathway, it can rise the protein synthesis, especially located in the neuron damage site. Next. Also, it can promote the neuron outgrowth. So you can see each dot indicate the one cell, the one neuron. So here we can see either DHEA or 179 overexpression compared with the control. The outgrowth and the neuron length is a significant increase than control. Okay, next. Also, we go back to the animal model to check the neuron expression. So similarly, we use a MAP to this neuron marker. We can find the 117 overexpression indeed in the damaged side. The expression level is higher than control. Next. And then here we use a different strategy just because DHEA is one kind of steroid. It is quite difficult to be a drug because it will metabolized by our body. So we try to use a compound block this interaction between the sigma-1 receptor and 179. This is an HDAC inhibitor. So next. Similar, we also get a similar result. HDAC is able to increase the neuron length and outgrowth. Next. So we also perform some animal behavior to confirm our result. Okay, next. So in the open field study, we check the HDAC inhibitor indeed it can increase the motor behavior, reduce the anxiety. Next. And we also increase the next building behavior because it's a kind of mouse healthy behavior. So this compound treatment indeed it can recover the building behavior. Next. As well as increase memory behavior. Next. So go next and the next. Okay. So here I just show you our model. When we got a brain injury, the axon or neuron will damage through the brain injury. And then the HDAC or some mechanism, it will block the 179 and sigma-1 synchrony allow the neuron, so the result just cause neuron cannot repair so well. But when we use the HDAC inhibitor or enhance the sigma-1 function through the DHEA, indeed we can trigger the protein synthesis as well as allow the growth cone of the neuron migration to the target region. Okay, next. So here I just share you some example for a topic in my lab, and then we study this kind of traumatic brain injury through the compound design, through the molecular experiment conduction. Also, we cooperate with neurosurgeon to collect some patient sample. Through the mechanism, we also found some potential target or potential biomarker. It could be the diagnosis mechanism or diagnosis approach in the future. Yeah. All right. Okay. Sure.
Before talk, we bring some of our TMU back for the students. In the end of my talk, I will ask some very, very simple questions. So feel free to answer the questions. Okay, Very happy to be here, and my name is Minhan Wu from the International PhD Program for the Translational Science in TMU. Okay. No. Don't too fast. So today I'm going to talk about the antibody drugs conjugates, especially in cancer therapy. I think maybe go to the next slides. I came to know i3L from next slides from 3 students from i3L, Erica, Eldra, and Celestine. In this summer, he joined us our program, and they are very hardworking students, and they are creative, they are talented, and they also visit a lot of places as well. And also we bring them to visit a lot of biotech company or any research institutes in Taiwan. So we hopefully in the coming years, more and more i3L students can join the T program, especially in TMU. Okay, next. Okay, so today my topic will divide into 3 parts. The first part is briefly introduce the research highlight in 2 programs. One program is our program, Translational Science program. And the other program is the undergraduate program, which is a medical laboratory bioscience and biotechnology. Okay, next. So, anything? No. Okay. So before we talk about the research highlight, we want to let you know what's the translational medicine. The translational medicine is kind of bridge between the bedside. Bedside is the laboratory to the bench side, to bedside is the clinical stage. So in the past, these 2 research are parallel line, they never connected and never talk to each other. So it is about these 20 years, people start to think about if we can connect this information together, maybe we can help the patients have better treatment. Okay, so in this kind of translational medicine, we need to have many informations. For example, we need to have a very best basic research to discover the diagnosis or therapy targets. We need to learn how to design the preclinical research, especially mouse study or other mammal study. And also we need to learn how to do the clinical research. And finally, even this product or use some commercial diagnosis kits, you want to diagnose the disease, how to make the decision, and how to do the clinical practice. Okay, so all of the effort is, please go to next, is to cross the Death Valley. Because if we connect it together, we can go from the bench and push the result to the clinical usage, or we can bring some problem from the clinical and then go back to the bench to do the research. Okay, next. Okay, so the goal is we, in these 2 programs, we create the translational platform for the clinical diagnostics. Okay, next. Okay, in our program, we focus on 3 aspects. One is the organoids. If you ever have the chance to culture the cells, usually we culture in the plastic dishes, right? So it's kind of 2D cultures. But nowadays, people know that the 2D culture is kind of not mimic to the real stage. So we focus on the culture the cell in the 3D 3D dimension system, and the more mimic the tumor microenvironment. And we have some spatial organoids combine the neuron and muscle cell to have a spatial co-cultural organoid system. And we have the preclinical model, we have the genetic mouse, modified mouse, and PDX is the patient-derived xenograft. And finally, it's a specialty in our program is we have the protein and structural biological lab. we can generate the monoclonal antibody, we can identify the protein structure, and we can help to design the recombinant protein drugs or peptide drugs. Okay, next. And in the medical science and laboratory program, because they have the undergraduate and they need to get a license, so their research education, they will have 5 aspects: medical laboratory science, precision diagnosis, microbiome pathogenesis, regenerative medicine, and immuno-oncology. Okay, next. Okay, I will show you some of the research results that is really high impact. We don't want to talk about the impact factor. We talk about the impact. So this paper published in the Journal of Clinical Oncology, they use our national healthy database, because in Taiwan we have national healthy system. We can have the database, and they found this antihistamine, this drug. When use this drug, they can reduce the liver cancer potentials. And these patients are the HBV, HCV, and the dual infection patients. Okay, so this is the clinical benefit for the clinical doctor in the future. If they want to treat this kind of patient, they can use these drugs, because these drugs is clinical available. Okay, next. And we also do a lot of effort in the COVID study. So in here you can see this paper mainly focused on the post-COVID infected patients, which means if the patients survive in the acute stage, how about the persistent and after the long-term COVID? Okay, so they find some immune signature, for example, the high TNF-alpha and IL-10 ratios. If you have this kind of phenomenon, which means they will have the poor outcome even after you survive in the acute stage. Okay, and next. Okay, another one from our department is we try to push all the basic results to the clinical use. So our program, Professor Yang, he generally he established this spin-off company, and her product is to diagnose the endometriosis. And she already got the Series A funding. She already got a product. She is doing the clinical trial in US and in Taiwan, and try to get the FDA approval and increase the market. Okay, so this is already switched from the basic to the clinical size and on the market. And the next one is we closely interact with the industry. So Professor Chen, she has the ability to generate the genetic modified mouse. So she collaborates with the industry to generate the disease model for COVID infections and vaccine development. And she also helped to design the immunotherapy based on some glycan-binding immunoglobulins. Okay, so these are the research highlight in our 2 programs. Okay, next. Okay, because today's topic is antibody drug conjugates, so I will briefly introduce the recent advance of the cancer therapy. Maybe I will talk about some of the history. In the new generation, maybe you don't know what kind of suffering in the past. Okay, next. Okay. During the past 40 and 50 years, you can see actually the cancer survival rate is increased double from about 25% to 50%. But this already takes 40 to 50 years. And next. But you see, this is the US President Biden. When he is the vice president, he already raised the funds for a project called Moonshot. Maybe many of the students know it. Moonshot is kind of starting off the precision medicine. Okay, so after he became the president, he mentioned to the media he wants to reduce the cancer deaths by 50% in the next 25 years. I think it will be achievable, because you see nowadays the biotech technology get progress very fast. So I think this is in the near future, we can see it. And later on, we will introduce the more modern therapy. Okay, next. Okay, so I think many of you know that cancer is a disease of DNA. So the mutation in the DNA cause the oncogenes mutations or the tumor suppression, inactivation of tumor suppressor genes. So they are driving force for the tumor, for the cell's abnormal growth. But at the same time, they generate several tumor markers on the cell surface, which could be targeted by some of the medical treatment. Okay, next. So before 20th century, all the weapons we can use is, for example, surgery, for example, chemotherapy. The chemotherapy is due to the World War. They found that the toxic gas have some element that could kill the people. So that's why they could start the chemotherapy. And the radiation and the hormone therapy for some hormone-sensitive tumors, such as breast cancer, prostate cancer. Okay, so this is our story between before the 20th century or next. But after going to the 20th century, the first target therapy has been discovered and focused on one type of a protein called the tyrosine kinase inhibitor. If you ever read the signal pathway, you will know tyrosine kinase is the most important signal transducer for the cancers. So the first tyrosine kinase was discovered. Next. Okay. So in 2001, in Time magazine, this is a breakthrough in the cancer treatment history, Gleevec. Okay. These tablets, these drugs, they kind of cure one of the cancer. Next slides, called a CML, chronic myeloid leukemia. Okay, so before these drugs, the survival rate of this patient is 30%, but in a clinical trial, it's reached to 90%. But this is just the first generation. They have second generation, third generation. So for CML patient now, kind of we can say this cancer is cured. Most of the patient can survive more than 10 years. Okay, but the stories is mainly focused on some leukemia cells. For the solid tumor, especially like this patient is melanoma, the story is not easy like the imatinib. Okay, next. After the target therapy treatment, you can see this is complete response. Tumors was gone. You were very happy and hope everything will be successful in the future. But next slide. The best thing is the recurrence. That's the weakness of target therapy is the recurrence, because you kill maybe 99% of the tumor, but 1% of the tumor will go back maybe 1 year or maybe 6 months or maybe 1 month. It depends on different patients. So the heterogeneity of the tumor, which cause the defects in the target therapy. Okay, next slide. So what's the next generation therapy or next magic bullet, okay, to the cancer therapy? Next slides. So nowadays, the antibody-based therapy is dominant in the cancer treatment. Okay, let's see the truth. This is the 10 best-selling products, the drugs in the market. You can see in the 10 best-selling products, half of them are monoclonal antibodies. Okay, the monoclonal antibody, they have their advantage is the specificity. The target therapy, small molecule, although they have the specificity for the kinase, but still they have a lot of off-target effect. So antibody is more specific compared to the small molecule, but the weakness is they only can target on the surface antigen. Okay, next. Okay, so this is the progression from the earliest antibody from mouse. Okay, This is the first generation, and second generation is chimeric. Chimeric is they retain this variable region, so maybe about 30% of the antibody from mouse and 70% from human. And then so as time goes by, new technology create the humanized antibody. The humanized antibody is to retain this we call CRD complement domain and to recognize the antigen. So in this humanized antibody, only 5% of the antibody from mouse and 95% from human. And now we have the fully humanized, fully human antibody. So 100% from human. So as the name you can see is from the most earliest, the MAP. MAP is the monoclonal antibody, to the ZMAP, to the ZooMAP, to the MoonMAP. So for pharmacy student, next time if you look at the name, you must know what kind of antibody you're using. And also, different targets have different names. You target on the tumor, you will have the TU. You target on the cardiovascular system, you have the CI. You have lympho system, you get LI. So this is the capture for the pharmacists. I think many pharmacists then you already know that. Okay. Okay, next. So use this antibody, the latest therapy was generated. The first one is the most popular one is the immune checkpoint inhibitors. In the immune system, we have the cytotoxic T cells. Then cytotoxic T cells, they can use the T cell receptor to recognize the tumor antigens through the MHC antigen presentations. But the cancer is very smart. They use another weapons called the PD-1. They will bind to the PD-L1 ligand from the cancer cells, then sending a negative signal to weaken, to terrorize the T cell, cannot recognize the T cells, okay? Cannot recognize the tumor cells. Okay, so next slide. Next. So if go back. So if we can block the interaction between the PD-1 and the PD-L1, then we can awaken the T cells and killing the tumor cells. Next slide. So next slide. So we have so many antibody as immune checkpoint inhibitor like the pembrolizumab, nivolumab, alizumab, and also the ipilimumab. This is also the Nobel Prize in 2018 from Dr. Hongzhou from the Japanese and Dr. Allison from the US scientist. Okay, so this is the first example. Antibody is so effective in the immune modulation. Next slide. And we have the first cell therapy also related to the antibody. This is called CAR T therapy. CAR T is the chimeric antigen receptor. CAR means they combine the antibody and the T cell receptor. So you can see here, they use the antibody as a monitor system to recognize the tumor-specific antigen. But this CAR still can transduce the T cell signaling. So after this CAR recognizes the tumor cells, the T cells can kill the tumor cells. So this is the breakthrough in the past 5 years. But cell therapy, CAR T cell therapy is very, very, very expensive. If you look at the website, you will know. If you want to do this, maybe, I don't know if in Indonesia, in Taiwan, we just approved it just this year or last year. But if you want to do this, it takes several tens or hundreds of thousands of dollars. Okay, next slide. Okay, so go back to our topic today is the antibody drug conjugate. Okay, ADC now is very powerful because they combine two aspects. One is the specificity of the antibody. And then another specialty is they carry out kind of chemotherapy. So they let the chemotherapy become specific, use the antibody. But the science of the ADC, antibody drug conjugate, also contain the linker, because we want this linker linked to the drugs very, very stable in the plasma. You don't want to let the ADC release in the bloodstream, because the payload, the drugs will kill not just tumor cell and also normal cells. Okay, so this linker must be stable in the plasma, but we want them to be cleaved in the tumor cells. So this is a design and a science in the linker. So there are 3 parts in the ADC. So antibody have diversity. Linker also have the diversity. Payload, you can have different kinds of toxic agent. For example, you can have the tubulin inhibitor. Tubulin is cytoskeleton. Okay, you inhibit cytoskeleton. And you can have the topoisomerase inhibitor to inhibit the DNA replications. So they have the diversity in 3 parts. So in the pharmaceutical company, they like this because they can have a different diversity and generate different kind of drugs. So as I mentioned earlier, target therapy, maybe you need to treat one drugs and fails resistant and then get another drugs, okay? And another special point is the DAR, drug to antibody ratios. The higher DAR then means more drugs carried by antibody. Okay, next slide. Okay, so the basic action of the mechanism of ADC is the antibody will target on the spatial molecule, but this molecule must be internalized into the cells. Especially they need to transport to the lysosome. In the lysosome, they have the caspacin, that kind of enzyme to cleave the linker and then release the payload. And then the payload can kill the tumor cells. Another specialty is the bystander effect, which means after the the payload released from these cells, and then they can kill the neighboring cells without this tumor marker. So even just 30% of the tumor have this marker, but when you treat ADC, they can clean all the tumor cells. So this is the power why ADC is so popular in the recently. Okay, next slides. Okay, I will give an example from the HER2. HER2 is a spatial tumor marker in the subtype of breast cancer. So the first drugs of HER2 antibody trastuzumab in 1988, and the second one is the pembrolizumab in 2020. But special thing is that in 2013 they have the first ADC. Their DAR is 3.5, which means they carry about 3 drugs in one antibody. But the most famous is in 2019. These drugs in HER2, if you look at the Google, you will find this is a very famous drugs. The AstraZeneca collaboration with the Saito in Japan. And they can generate antibody conjugate with 8 drugs. So it's very effective. Next slides. Okay, so you can see here, they improve the survival rate compared to the first generation ADC from about 7 months to about 30 months. This is a big breakthrough. So in HER2 now, not just treating the breast cancer, any cancer with the HER2 overexpression, you can treat it by this in HER2 and ADC. So ADC is very powerful because, as I mentioned, they have a bystander effect. They can kill all the tumor mass. Okay, next slides. Okay, so currently, already in the market, we have 14 ADC drug got approval and more than 100 ADC in clinical trial. And the market size is about 10 billion and estimate in 2030 is about 15 billion. So this is a good field. And if in the future you want to join the research, this is a good topics if you want to. Okay, next slides. So I will give some time to talk about some of my study is focused on an anti-6 heavy chain antibody drug conjugate. Next slides. CKN6 is a GPI anchor protein. Is the GPI anchor protein? Is the protein that link using the glycolipids. So they only anchor on the outside of the cell membrane. They don't have the transmembrane domain. They cannot transfer the signal, and they are highly glycosylated. And the special thing is that could be the tumor marker. The most famous member in this family is the CKN5, which also can call CEA. If you have the colorectal cancer diagnosis, CEA is a very good marker in a clinical practice, okay? And CKN6 is another member in this family and also a good tumor marker. Okay, next slide. So we generate the antibody, next slide. So we generate a special antibody from an animal called a camel, one of the camel called NAMA. This antibody only have the heavy chain. Has the heavy chain only antibody has the advantage in the size, because you can see conventional antibody, you have the heavy chain and light chain, and the size is much bigger. So we have smaller size easily for the tumor penetrations. Okay, second part is that because it is small, so they can find some special epitopes on the cell surface. Okay, the conventional antibody, because much bigger, sometimes they cannot find some epitope that heavy chain antibody can bind. And another advantage is the small thermal stable. Thermal stable means that this antibody you can put on just a bench for 1 month, still get a very good affinity for the antigen. Okay, this is the advantage in this heavy chain antibody. Next slide. Okay, so we use this antibody and take out the tumors from the oral cancer patients. As you can see, upper panel is the heavy chain and lower panel is conventional antibody. You see they have a higher affinity for the T is the tumor, N is a normal, have a higher affinity to recognize the tumor tissues. Okay, next slides. And we confirmed, we confirmed this antigen recognized by the heavy chain antibody is a glycosylated antigen, because when we treat enzyme to remove all the glycan from the protein, you see the size will dramatically decrease, which means the glycan already removed and they can recognize the antigen. Next slides. But why the conventional antibody has poor affinity is because in here you can see these are the samples that cannot recognize by the conventional antibody. But if we remove the glycans, you can see the antigen could be detected, which means the glycan in the protein, they can mask or change the conformation that conventional antibody could not recognize. So our antibody has the advantage to recognize the glycosylated CKN6. Okay, next. So use this antibody, we see in the tissue they have very good staining, and if the high expression, they have the poor recurrent survival. Okay, next. And we demonstrate this antibody has no affinity to the normal tissue because we wanted the specific for the tumors. Okay, so you can see most of the no matter it's colon, liver, lung, very low expression level. Next slide. But one special thing is they express in the granulocyte in the spleen, which means they may have some toxicity for the white blood cells. But later on, we'll show you there's no toxicity. Okay, next slides. But go back to the tumor cells is totally different. You can see no matter is the colorectal cancer from rectum, descending colon, or they already metastasized to liver, or lung tumors, cholangiocarcinoma, they all got very, very high staining, which means they have very high specificity for the tumors, this antibody. Next. Okay, and we demonstrate this antibody is spatial, but we want to know whether CKN6 have any pathological effect on the tumor progression. So we overexpress the CKN6 in the oral cancer cell, and the cell will become more invasive and higher mobility, and they can grow faster, and they can metastasize. This is the 12-vein injection to see the lung nodule. You can see more lung nodules in the overexpression cells. And the mechanism is this CEACAN6, they will interact one important molecule called EGFR. EGFR is the epidermal growth factor receptor. So if you do the cancer target therapy, you know this is a very strong signal for tumor growth. So CEACAN6 interact with EGFR and promotes the EGFR signaling and cause the cancer malignancy. Next slide. And also not just in oral cancer, in colorectal cancer is the same. We took out 2 overexpression clones, and you see in these 2 clones, their EGFR signaling is much higher. Next slide. The important is to prove in the animal models, and cetuximab is anti-EGFR antibody. So we combine this antibody together. Although you see along the green one, this is the heavy chain antibody, is not effective. But when they combine this red line, you see they much enhance the cetuximab effect, which means if the patient got cetuximab resistant, maybe we can add the heavy chain antibody to increase the therapeutic effect. Okay. But this is not enough. Everyone wants to know once the tumor gone, tumor free. Okay, so next slide. So when we apply the grant from the government, the reviewer told me that maybe you can think about the ADC. Okay, so we collaborate with one institute called the Development Center of Biotechnology, DCB. They have a special technique in the linker technology. They use the glycan. This glycan we call the trimanose. Okay, so use this trimanose linker. One antibody can conjugate with 4 drugs. Okay, so use this platform, we generate our heavy chain antibody ADC. Okay, next slides. To see is to believe. So you can see this red line. When we generate this ADC and inject to the tumors, you see after 3 injections, tumor was gone. And the effect could last more than 2 months. And even in the end, when we sacrifice the tumor, when we open the mouth and the skin, there's no tumor. So that's why ADC is so powerful. That could be the next, we said, the magic bullets for the cancer therapy. And the yellow one is the clinical regimen for the cancer, for the colorectal cancer treatment. So you see in the traditional therapy, although they can suppress the tumor for 8 weeks, but after weeks, the tumor become resistant. Okay, so in the future, if the first line therapy fail, ADC could be the second line, or maybe in the future, they could be the first line therapy for different kind of tumor. Okay, next slide. Okay, and we demonstrate this theory in different kinds of tumors. So we see this antibody can recognize different kinds of tumor, colorectal cancer and head and neck cancer and the leukemia cells. But their affinity to some cancer is low, or they have about 10% in the PBNC. PBNC is peripheral monocytes or mononuclear cells. Next slide. And we prove that this antibody actually will be intact into the lysosome. We use the lysosome tracker and stain the antibody. They colocalize to show the yellow color, which means the antibody could be endocytosis to the lysosome. Okay, next slide. Okay, and we demonstrate the specificity. We overexpress the CEACAN6 and 5 in these H1299 cells. And you see in the red line, only the CEACAN5 cell get very good cell killing effect. But in the CEACAN5, their effect is not very strong. And we also use another liver fibroblast as a negative control. You see kind of no effect on these cells. Okay, next slide. And another one is surprising us. Remember the PBMC, we have about 10% of the cells have the CEACAN6. But when we do the cell viability, you see kind of no toxicity in the PBMC. But another thing is THP1, just only about 30% positive rate, but the ADC could kill all the leukemia cells, which means maybe the bystander effect exists in our ADC. Okay, so these are the current study in the antibody drug conjugate. Okay, next slide. But think about this is ADC is a very complicated product. You need antibody. Antibody is the biologics. You need the linker. You need the chemotherapy drugs. These are chemistry. Okay, so if you want to do everything by yourself, I don't think it's possible. Okay, so next slide. So now on nowadays, usually if you want to do the translational research, you need to have a team. For example, if you have the product, you have the prototype, you will have the CDMO or CMO. The CDMO is that from the beginning, for example, ADC, you need to have the cells to produce the antibody. So you may need the company to help you to select the cell line, generate the antibody. You need to have the company to help you to conjugate the drugs with the antibody. So it's a teamwork. And nowadays, especially now the COVID, when they want to produce the vaccine, they also have the CMO because they cannot produce by one factory, okay? So nowadays, we need to have the teamwork, not just for the ADC, for many biologics. Okay, next slides. Okay, so CDMO is the one problem. So if you would want to translate needs money. Okay, everything needs to start from the money. Even professors want to do research, they don't have money. Okay, so but for the to start up a new company, you need to raise the funds. Okay, so it's about to end this talk. So I will have some questions for you. Okay, next slides. Where get the fund? We always say 3F. Okay, 3F. Can anyone tell me what's 3F? You can Google it, or you can directly say, raise the fund from 3F. Okay, it's a very popular word when you want to commercialize for your drugs or your product. Okay, we may have 10 or 20 minutes. We will allow 2 to 3 students to answer the questions. Okay, anyone find the question and find the answers? 3F? No? That was surprising. 3F. 3 words start from F. Anyone want to try? Oh, yes. Yes, 3F. Like, next slide. We will give you the bags, but you already answered 3, so only prepare one question while you finish all the answers. Okay, next slides. Okay. Next. So, friends, family, friends, and fools. But don't think fools is fools. Fool is because they trust your research. They trust on you. So kind of they addict on your research. So they whatever you say, they trust you. So they will invest in you. So don't think it's an idiot. Okay. So 3F is very important for you to raise the first fund for the commercialization, especially ADC is very, very expensive if you want to commercialize. Okay. Okay, I will end up in one word. Next slides. Okay. Every time when I talk to, this is real, when I talk to my students or any topic about cancer, I always show this slide. I can survive. If you have the relative or friends, they are suffering from cancer, always tell them. Because new drugs coming out year by year, month by month, so supporting them, that then can sustain. There was hope in the future. Okay, So thanks for your attentions, and these are the family or these two programs, and we will welcome you and more than welcome the students from i3L to join us. Thank you very much.
Next up, we have our third lecturer, Dr. Chi-Yang Wang. The floor is yours.
Some students not come here, so I can hear some. Okay, thank you. Okay, so good afternoon, everyone. So it is like the times still have some limit, yeah. So I will try to turbo my talk, yeah. Hopefully not to delay the next schedule to everyone. So my name is Dr. Wang from the Department of the Cancer Biology and also Drug Discovery. Today, I would like to share my introduction from our department, and also I would like also to share some topic from the application. Okay. Okay. So how you did you? Yeah. So. This one. Yeah. Okay. So today, after introduce our department, I would like to give a talk about my currently research. Especially, we will focus on the application of high throughput technologies in cancer biology and also drug discovery. So this is my outline. In the beginning, I would like to give some brief introduction of our PhD and the master program of cancer molecular biology and also drug discovery. Later on, it's the Greater Institutes of the Biomedicine Informatics. Finally, I would like to introduce currently my research focus on glioblastoma, especially to show how to use the high throughput technology like the single cell RNA sequence to solve the glioblastoma intratumor heterogeneity. And also, I will also share some experiments about the tumor microenvironment, especially targeted in the breast cancer. So first, will be the introduction of my department. My department, they are not only consistent of the domestic students, but also have the international students, including the master and the PhD degree. The students is all over the world, not only in the Indonesia, also from the Milan, Thailand, and also Africa countries, and also United States. Yeah. So when students come here, they can also learn different cultures and also learn a different skill from the classmate. And as the Bastien talking about, we also provide the type A and type B and the type C scholarship. And over that, they will also have the tuition waive. Yeah. So students can also totally focus on their research without the life warrior. And our department, they will be focused on the biomarker identification in the beginning, and also it's called the basic research. And later on, when we use this approach to find some biomarker, we will also combine the omics-based targeted therapy, targeted identification. Then later on, we have a very strong teams. It's the AI-based drug discovery and also new drug discovery and also drug repurposing. It's also especially leading by our teams here. And all of this, they will also facilitate the drug development. And finally, we also have some several cases already be cooperated with the biotech company, and some of them they also are undergoing, and hopefully some of the drug could be go through the clinical trial and also phase 1 to phase 3 trial. Yeah. And we hope in the future they will be have some new drug will be developed. Second is like we would like to introduce the medical informatics department. This department also consists of the master and also PhD program. This program is especially targeted on the medicine, pharmacy, nursing, information, and also medical engineering. And this department, they are majority analyze the biomedical information like the X-ray or some CT, MRI, some image, and use the AI technology like deep learning, transfer learning, and also some machine learning, the other algorithms. All of these tools are all facilitated to promote the precision medicine, because to deal with this huge data, it's quite important. Yeah. So if anyone in the bioinformatics department is also welcome to join for this program. And this program is very productive and also have a wider cooperation, not only in Vietnam, but also in the United States and also different countries. So before, if you not decided yet whether want to come to Taiwan or not, they are type of program. So after this presentation, I will also show you some movies. They will also show you how the previous students, and how they dedicate their work in the science, but also enjoy life in Taiwan. So anyone, if they are interested, they can also scan this QR code, and we will have a further one-by-one discussion in the future. And also in the meantime, we also have our fall admission already be announced. So everyone, if you almost finish your bachelor degree, you are also welcome to apply our master programs. This application will be from November 15 to next year March. So if you get the admission, then you can start your master program through the next year September. So overall, the previous slide just show you the background and also some information to our cancer biology department and also the medicine informatics department. Then the second, I would like to introduce my recently studies, especially focus on in the beginning, I will show you why we want to study the glioblastoma and why we want to use the single cell technology to solve this problem. And we are including the motivation, methods, and how we analyze this big data and how to combine these bioinformatics data structures. And finally, we are also showing some tumor microenvironment in the breast cancer. So first of all, I would like to introduce the background for the glioblastoma. So why we want to target in the glioblastoma? Because the glioblastoma is the common brain tumor, especially they occupy almost 48% of the malignant CNS tumors. And almost it's also the top 10 cause of cancer related deaths in Europe and also North American. And why we want to target this and why it's also a challenge for this glioblastoma. So in the A panel, we can see, usually we use the surgery, we can only take out the red colors. We can also call it the tumor core, or it's the majority of the tumor mass. However, if we take out the tumor mass, so as you can see in the middle picture in the B, the tumor mass already be removed by the surgery. However, but still have some green and also the green color and also the blue colors. This is surrounding, we call the tumor margin pass. And this tumor margin pass will be day by day and year by year to lead to the tumor recurrence. So this is very important to understand what's the detailed mechanism and how to acquire and also kill these remain tumor cells to avoid the tumor recurrence and also metastasis. So this is the pictures to show you, as you can see in the middle, the 4 centimeter is the tumor core, and the outside surrounding 2 centimeter is the tumor margins. So although we take out the tumor core, through the surgery, as you can see, the ratio to the tumor cell and the total cell is like a one by one. However, the rest cell, even they are 1 to 100 or 1 to 1,000, although the population is very low, but in a long time, they still have to be possibly to recover the whole tumors again. So this is the brief introduce why the tumor usually when we take the surgery, but in some day they will also recover again. Yeah. And to understand this mechanism, we needed to know actually in a tumor cells in a brain, they are it's composed of lots of like a stroma cell, not only the tumor cell. As you can see, they still have some the other like a T cell, NK cell, macrophage, and also kind of like the fibroblast. And all of this, they will also establish and also create the environment to facilitate this cell, to promote the tumor cell regrowth and also active again. Yeah. So some people may also ask some question why they don't want to take out all the tumor mass, because in a tumor brain, in a brain, the tissue they are control multiple biological functions. So if we take this surgery, take too much amount of the tissue, they will also cause a huge damage, to the human functions here. And in the past, the TCGA, they use the for like a different subset, for different subset, and also use the several markers to do the identification. However, later on in the cell papers, they also use the OPC, MPC, AC, and the MES markers to illustrate actually in a tumor, they are highly heterogeneously. So if we just use the traditional like a TCGA, it's like the bulk sequencing, or you can call this also kind of like the traditional high throughput technology, including some like the microarray or RNA-seq. But all of this layer cannot to be well to answer the tumor heterogeneously. So later on, I will also show you why this is very tough and also a challenge to use the traditional high throughput technology to solve the genetic regulation. So the rationale is like, as we discussed before, the glioblastoma recurrence is almost happening in the tumor margins, especially in the tumor restation paths. However, detailed mechanisms still are not be clearly be defined. And also currently, the glioblastoma biomarker is also not enough to answer this question. So therefore, we use the single cell approach to investigate the crosstalk between the stroma cell and also tumor margin in the glioblastoma. So why we want to use the single cell technology to solve this problem, and why is the motivations? As we can see, you see the in the left panel, when we do the traditional high throughput technology like the microarray or the RNA sequencing or proteomics, actually they just get the average gene expressions. It's kind of like you put all the fruit together and then make it kind of like the fruit juice. So all the signal and all the gene expression, they are mixture to each other. So this technology is good, but it's better to study the homogeneous disease. However, using the single cell technology that we can wear to to separate like a banana and also cranberry and also grape. Yeah. And this is very important to study the heterogeneous disease. As we show in the beginning, the tumor are either in a tumor core or tumor margin in a brain tumor. They are heterogeneously. So they are composed of lots of like the not only cancer cell, but also the immune cell, even the cancer stem cell. And as we all know, the cancer stem cell is very powerful and also dominant for the cancer drug resistance and also the cancer recurrence and also metastasis. So this picture is more easy to help everyone to get this concept. It's also we use the left panel picture to remind you again. In the tumor trunk, tumor mass, they are not only the tumor cell, but also have lots of like the immune cells. And all of these immune cells, they will also promote the nutrition and also secrete either the paracrine or autocrine, some growth factor, and also some cytokine to promote the tumor growth. However, if we just use the traditional microarray or RNA sequence to acquire for this tumor cell and also this whole tumor trunk, then we just get in the right panel, the AVG signaling of the whole like a tumor population. This is good, but also they will also lose and also ignore the real cell, especially for the cancer stem cell. They may only have the 1% or even 3% for this in the tumor tissue, but they are really dominant for the cancer development. So in the later on, I will also show some slide to show you why this is also important even they are very rare cell, but they can also cause for the cancer resistance. This is a very traditional and also like a very straightforward picture to show you this concept. So in a up panel, as you can see, if we use the bulk sequencing to analyze this tumor trunk, as you can see, in the tumor, they are composed of like different types of the stroma cell, like the cancer stem cell, immune cell, and also the other stroma cell. However, if we just use the bulk sequencing and do the RNA sequencing or the kind of like the microarray, then we just get the AVG signaling, as you can see, the red population cells. However, if we, according to this gene expression, design some chemotherapy drug, and then only the red population cell will be killed. However, as you can see, in the long run, the red and the blue and also purple population cells, they will still survive because the drug are not target for this subclone. Then, after a couple months or even a couple years, the tumor will be recurrence again. However, if we use the single cell RNA sequencing, they will be totally different. We can also, according to the different subclone, the different population cells, then get the individual signaling. Then, according to this individual signaling, then we can design, the medical doctor can also design different chemotherapy drug to kill for this cancer. Then, finally, all the tumor cell will be killed, and also tumor will not recurrence again. So this is the idea why we use the single cell technology, to do this research and also try to find some drug they are targeted for this important cell. Even they are very rare population, but they almost dominate for the cancer cell recurrence and also metastasis. So when we know the importance about a single cell RNA sequencing, the basic knowledge, then I would like to introduce some methods. The methods usually in the beginning, as we can see in the left panel, we acquire for the tumor tissue or like any tissue, either from the hospital, from the human, or from the mouse. Then we can use the in the middle this panel, use the tubes or the microwave or microfluidic or like a barcode system to separate this single cell. Then later on, we can send this cell into the sequencing machine. And finally, we will rely on bioinformatics guys, to analyze this signaling. And finally, we can get what kind of like a drug could be useful to target for this different population for the tumor cells. So this is the history from the withTaipei and also the microfluidic and also the pico wheel and also the barcode system. And each system, they have the pro and the cons. Yeah. So it's also very important that to understand what you want to study. In the beginning, some people may use the flow cytometry to sort in the specific population to the single cell. But finally, in the current stage, most people will rely on the drop layer system. And also later on, I will also show you a very powerful system. They call the 10S system. So the 10S system is also called the single cell technology genomic systems. They use the barcode system and also help you to acquire and also label different cell population. Then finally use the computer science tools to help you to separate the signaling from the different individual cell, like the cancer cell, immune cell, stroma cell, and also the fibroblast. Then medical doctor can also, according to the signaling, design a different drug, and hopefully this drug can also cure for the whole cancer recurrence. And later on, recently, some company, they also designed the BD recipe system. It's also combined the like flow cytometry and also the sequence genomic systems. Yeah. So this will be also can double confirm and also can also save lots of like cost of this reagents. So when we get this single cell data, how we can analyze this data. So as we can see, we get the tissue from the hospital, and also we use the different tools to to separate the single cell. And finally, when we get this high throughput database, then we need to rely on the bioinformatics department students, to analyze this data. So the most powerful packages, like we use the cell arranger and also Shure and also this single and also the monoclonal package to analyze these data. So when everyone, if they do the presentation, the precision medicine and also the single cell technology, they will be they will be very familiar with this single cell system. And most of all, they will use the Xu to do the identification and also the annotation for different population of the different single cell. And according to this single cell, they can design for a different drug to treat for the tumors. And also the other package we call the monoclonal. It's kind of like the trajectory and also the cancer evolution system. So through this, we can also easily to know what is the cancer initiator and where they are come from and how they can develop to the cancer recurrence and also the metastasis. So this is just like we give you some brief introduce because due to the time limitation. So I only give you some kind of like a very simple concept. But if you have any interesting, I think we can discuss later or even join our tip program so you can learn how to analyze these data. And also, of course, there are also some different packages. They help us to do some the other epigenetic regulation and also to check what kind of like the transcription pattern, they will also regulate the upstream signaling. This is also very important for we to know what kind of like a gene or what kind of like the transcription pattern they are dominant for this signaling. And all of this, we can also easily to design for some drug to like to block the upstream signaling. So we will also share some like we read some publications. Yeah, the first is like we cooperated with the Bastin as he talking about here. His paper also focused on the glioblastoma studies. So in the last year, we also follow like his talk later. We find some HTAG inhibitor. They can also regulate the non-narcotic RNA, microRNA, and so mRNA network, and to attenuate the cell growth and also preparation in glioblastoma cells. So we also see the non-narcotic RNA, 00461, they are also specific expression in the classified cluster. And also we also find that some drug they can also cure, for the has some benefit for clinical glioblastoma patients. So this is some cartoon picture. Anyone, if they are interested, they can also looking for our teams for the recent publications. And also the other parts that we are not only focused on the glioblastoma, we also focus on the breast cancer. And this paper is more focused on the how the mechanism and how the neutrophil they regulate for the tumor metastasis, especially in the breast cancers. So we use the two models. We call the HCI 001 is the high metastasis, kind of like a PDS models, and the other is like we call it like a low metastasis PDS nice is a 002, and then we acquire when we do the surgery to injection this tumor cell into the tumor mice, then we just acquire this tumor trunk to the signal cell sequencing. Then we find that the signaling dramatically regulated and also highly expressed in the tumor metastasis models. Yeah, and then we propose models. CCL2 they will regulate this neutrophil, and all of this they will also influence the tumor microenvironment and also lead to the cancer metastasis and also moderate the immune response. So this is kind of like we apply for these high throughput technologies in our current study. And we also have not only do the breast cancer, but we also do some colorectal cancers. And also this paper also published recently. So currently, our laboratory ongoing project is like we analyze the public database from the NCBI-GEO and also the single cell portal. It's very famous for the single cell technology related database. And then we try to find some potential biomarker and also go through the GSEA and also meta-core analysis to find some powerful regulation molecule and also the signaling pathway. And all of this, we will use either the cell culture or the mouse model to validate our current studies. And in our laboratory, we also have the other teams. They are focused on use some AI and also machine learning model to do some prediction. Because the single cell is very huge data. Usually we need to calculate thousands thousands, million, or even million sales, and also the million signaling. So how to use this AI, like the deep learning and also the machine learning tools and this algorithm to make some prediction and find some biomarker is also our aim and also our goals. Yeah. So this is also part of our currently research, yeah, in our teams. Okay, so I would like to thank you for all of the international students. So as you can see, there are lots of like i3L students, like the Andrew, Saba, Biona, Davy, and also Toby, and also like the Erica, and also Lonely. I think most of them you may also familiar with to each other. And in the final couple minutes, I will also share some movies and to let you know how they feeling their enjoy life in Taiwan. Yeah. So let me try to find the file. 要放吗？就是放个1分钟的影片。fine. Sure. Okay. Share screen now. Yeah, they may have some technical issue, but of course we will share this movie. Yeah, so everyone, if they are interested, they can also come here to see. Because there are some TIP students, they do a very great job in our TMU. Yeah, and they also make some very fantastic movies. Yeah, but I think we will try to solve this technical issue later. But we will start to move to the next session. Yeah, for the Q&A session. Yeah. So I can this stream video in the desktop. And so maybe they can share the movie later.
Oh, okay. Wait a minute, I need to share it again with you.
You'll always be my day one. Day zero when I was no one. I'm nothing but myself. You are no one else. Thankful you're my day one. Thankful you're mine. I got lucky finding you. I won't make the day that I came across you. 'Cause when you're with me. I don't feel blue. Not a day goes by that I would not redo. Everybody wants to love.
Yeah, that's right.
It's easy when you try hard enough. Yeah, that's right. You'll always be my day one, day zero when I was no one. I'm nothing by myself, you and no one else. Thankful you're my day one. I'm thankful you're my day one.
Okay, so this is the engine. It's a very amazing and also fantastic students. Currently, he finished the TIP program and also joined our master program in the cancer biology and the drug discovery. So if anyone have any question, yeah, I think you can also contact him about the life and also in TMU, not only focus on the science, but how to also balance and also enjoy life in Taiwan is also very important thing. Yeah. So anyway, for any question, we can still have a discuss later. Yeah. Okay. Thank you. Yeah.
Okay.
Request that everybody stay in to your house. Shoot them in the head to want to move the head from the brain.
I don't wanna go home today. You don't really wanna just go. The blue skies are telling me to go that way, yeah. I think I might just fly away, leave all my things behind, yeah. I think I might just leave the world and go run into my mind. Little blue flower, sing a little louder, get me a little higher so I can just dance with you. Little blue flower, sing a little louder, get me a little higher so I can just dance with you.
Okay, thank you so much to our esteemed guest lecturers, Dr. Jian Ying Chuan, Dr. Ming Heng Wu, and Dr. Chi Hyang Wang for the lecture. We now proceed to our Q&A segment. Due to the limit time, let's start with 3 questions from the live audience and 3 questions from Zoom. Let's start with the Zoom first, maybe. Is there any questions? Not yet. What about from the live? Our students here? Okay.
I have questions for Dr. Chuang about the nerve regeneration for a traumatic brain injury. I want to ask whether this treatment can be used for a comatose patient, for patients that are under coma. No. Coma like a vegetative state. How do you say it in English? No. The vegetative state, when someone has severe brain injury, they tend to like sleep for long time. It can be maybe. Yeah.
I think maybe we can measure about the disability for some spinal cord injury for example, some patient they cannot control their body, but the brain still well. For this kind of patient, maybe nerve regeneration is a way we can treat. But for the spinal cord injury, actually we have to solve the problem for the gliosis. Is a kind of a scar in the spinal cord. And then this kind of scar, it will affect the neural regrowth because it's a kind of condensed tissue. It will block the neural regrowth. So I think for neuroscientists, we try to treat for the spinal cord injury, allow the patient can walk again. Okay, for you said like a coma. I forgot how to say this kind of patient we call is in Chinese is just plain people. I'm not sure how to say this in the medical science, but yeah, for the coma, I say some patient they cannot talk or respond for the outside, and I think we also need to think about this not only for the neurosurgical function. We need to think about the consciousness. The consciousness is a kind of a problem. How we can let the patient wake up again. So it's not only the damage from the neural circuit, but also has a lot of function for example, like a computer. If the computer is still well, but you didn't get the software or app, I think the computer is no function. But the line, the electric, I mean, each part, they still well. So this kind of patient, just like the computer, if you lost some function, maybe you just try to like the line link again, but without the app. I mean, the computer dead without the app. It's so difficult. So it's a different kind of issue. For example, like a neurodegenerative disease or psychiatric disease, we are totally say they are totally different kind of disease. Yeah, so I just would like to let you know for the disability for the spinal cord injury or brain injury, I think we can solve the problem through the neural regeneration. But for the coma, a lot of issue we need to discuss. Yeah. Okay, thank you. But it's a quite good question because yeah, for neuroscientists, it's still difficult to treat this kind of patient. Okay. Yeah, thank you.
Thank you. More questions either in Zoom or in live? Okay, let's do the live one first.
Hello, I have questions for Dr. Ming Heng related to cancer cell therapy. Why does antibody drug conjugate have high tumor selectivity? And how does the HCAB or heavy chain antibody with ADC can prevent or reduce the toxic side effects on PBMC related to the side effects? Thank you.
I firstly answer the questions, the specificity. For the biologics, because each antibody, no matter is the heavy chain or light chain, they have 3 CDR. They use the 3 CDR to recognize the spatial epitope of the antigens. So that's why antibody, they have the advantage for the targets. But for the small molecule, usually we use the docking. For example, if this enzyme, they have some active sites, we use the docking to have the chemical simulation and fit to the active site to block this conformation structure. But somehow, a lot of protein, they will have a similar structure. And the small molecule is pretty small. So they may target on different kind of protein with some different affinity. So that's the something is good in the small molecule, something weak in the small molecule. Also in antibody is the same. And the second question is the heavy chain antibody has a better what kind of ability? Oh, this is actually the problem I should say this is the question we are also asking, because although they can bind to this PBMC, maybe just 10%, but we don't know why the cells still survive very healthy. And one possibility is also maybe the epitope exposure for the PBMC and the tumor cell maybe a little bit different. So maybe they bind to this antigen, but their still the affinity is not good enough, because in a flow cytometry, we will cut in a threshold. So no matter it's a very high expression or low expression, once they pass the threshold, we think this is a positive cells. So in the PBMC, maybe still is the low expression of CCAN6, but they already passed the threshold. So we found about 10% is positive. But that kind of low expression cells, maybe ADC won't kill them. This is just one explanation for the current study, but the basic study is still going on. So later on, maybe we can answer the question more detailedly. Thank you. And our vice team, they prepare some gift souvenir for students, ask questions and answer the questions. Okay.
Thank you so much. One more question from the audience, and then we have 3 more questions from Zoom. One more question. Okay, any more from Zoom?
Okay, so do the stage of cancer affect the treatments that were presented, like the T-cell therapy ADC? Example, how is the T-cell therapy implemented on survivor that have, say, stage 2 cancer versus survivor that are in the more later stages? I think this is for Dr. Ming, for you and for Dr.
Oh, yeah, both.
Dr. Wang.
I'm not quite familiar and know the questions, but I would say usually for this kind of systemic therapy is for the metastatic patients. Usually even because we always say if the tumor could be surgically removed, that's the good things. For example, the solid tumors. But once they already metastasize, so you will have so many weapons, T cells, CAR T cells, and immune checkpoint inhibitors, okay? So in the late stage, you will use the systemic therapy. But if they still in local, then we just do the surgery to remove it. So if you hear about even your tumor is huge, but they don't metastasize, you will feel safe because we just remove it. So that's the answer for, I think this is the first question.
Is there any more question from you guys? Okay. One last question.
So I have a question for Dr. Wu. So you mentioned about the CAR T cell therapy, but I was wondering, could you apply this CAR on other lymphocytes maybe, or on NK cells?
Well, you are very smart. Actually, this technology already used to different kinds of immune cell like a CAR, NK, CAR, macrophages. but the most difficulty is you need to find a good tumor marker. Yeah. Mostly now the current therapy of CAR T is focused on CD19. It's a spatial tumor marker in the B-cell leukemia. But in other solid tumor, although there are many clinical trials focus on the solid tumor. In leukemia, I will say CAR T is very effective. But for the solid tumor, I hear about the news from China. They say their CAR NK is very effective to cure. I cannot remember what kind of solid tumors, but I will put a question mark in this kind of research because solid tumor, they have very difficult problem is the tumor penetrations. If the tumor is a big mass, you want to let the antibody that the cell going to the penetrated tissue is not easy. So I will put a question mark in there. But for your question, actually all the lymphocytes, they are doable. And one special T cells now called a gamma delta T. There's another field because there's no immunogenicity of this kind of T cells. So we don't have to use our T cell because T cell, you need to extract T cell from yourself. But gamma delta T, you may use the T cell from other patients or donors and do the cell therapy. So this is the information that I can provide to you. Thank you.
Okay, thank you so much. After this, we would like to proceed with the with our token of appreciation. So if our lecturers could please come to the front. Oh, Erica?
Yeah, can I invite Erica to give us some?
Oh, sure.
Yeah, sure. After we take a picture.
Yeah, sure.
Okay.
Oh, Sir Rio, can you also join us, please? Sir and Sir Fandi also? Come on. So this is a little token of appreciation from us. Thank you so much for coming and giving us a lecture for today. Okay, and lastly, we have our own, we have before this, the certificates, yeah, certificate of appreciation. Okay, thank you. Okay, thank you. And then lastly, we have our own student here who has actually attended TMU. Erica, if you would come up and share your experiences. Okay. Oh, sure, of course, we'll share it in the email. The meeting and the lecture, yeah. Sure, of course. Will do. Thank you.
So hi everyone, my name is Erica Marceline Kwan. I'm a student, a final student here, a final year student from Pharmacy 2020. So I did my internship at Taipei Medical University just a while ago, 2 months ago, from July until August. And I see here that a lot of you are in your third year as well. So some of you might also be thinking what to do next. And like where should I go? And so I'm here to share my experience from the T program. Okay, so where did I find this program? So here is the link. You can also click here. And there are several other programs as well in here. And if you click on that, next news, you can find different programs. And last time I chose the medicine and public health, which I came across the next one. Oh, these are the types of the programs that you can actually find in the T program website. And next. And last time I did my internship program at the TMU. The program name is the Interdisciplinary and Translational Medicine Internship Training Program. So I did that program for 2 months. And what to expect? So there are 3 types of program, I guess, that you can expect by joining the interdisciplinary and translational science. So first of all, as you may have noticed, this is Professor Ming Heng Wu, the director of the translational science. So in that intern program, we will be given some professional lectures, and we can also have the laboratory internship. And also we visited some industrial and biomedical institutional. Here is the picture of us and my friends over there also, Celestine and Andrea. We visited the National Synchrotron Radiation Research Center. And we actually visited other places, but I can't show all the pictures. And this is the internship project that I did. So I had the honor to work under Professor Ming Heng Wu. So this is the title of my internship project, the investigating also the anti-CCAM6 heavy chain antibody on the pancreatic cancer cells. So what can you gain from this internship program? Global connections. You can make friends from Vietnam, Malaysia, Nigeria, US, UK, and also Indonesia. We actually have one friend here from i3L as well. Her name is Rachel. And here we have several Vietnamese friends as well here. And here's from Malaysia, from US, from UK. And here are the professors, very wonderful professors. And also we can get some cultural exposure. We visited many places actually. And here's the example of it is, if I'm not mistaken, is the Chiang Kai-shek. Yes, that one. It's very beautiful. And next, what can you do in Taiwan? A lot of things. We visited Hualien, we visited Keelung. And next, we also visited some beaches, the Waiao Beach and the Baishawan Beach. Marvelous. So to sum up my internship, it was actually a very memorable experience. Not only did I gain professional and also personal development, but I also made some new friends. I get to meet Professor Ming Heng Wu and other professors as well. And that's it, I guess. So don't forget to click on the link here so that you can see the internship program. Thank you.
All right, thank you so much. And thus concludes our Power Talk and our info session for today. Erica, don't leave yet. We want to take a picture with you. Okay. And also professors, if you would come up again and so we can take a picture with Erica. Yeah, it'd be more fun now. And also Pafandi and Saria, please come up also again. Sir Fandi aja? Okay.
Who's your mentor here?
This is the HOD, but the HOD is here. Okay. Okay. Come on, join up, join up. Let's go. Okay. Yeah. Okay, and lastly, can we get everyone here? And lastly, can we get everyone here, the participants, to the front so we can take a picture together? Let's go. So, the mana photographer kami untuk bisa ituin nggak atur-aturin fotonya? Oke. Okay. Thank you. Thank you, everyone. Thank you for making today happen. Okay. And that's okay.
Yeah.
We'll keep it for them.
Yeah.
Thank you so much. Thank you for today. Okay.
Students,
Some vouchers, if they are interested about EMU.
oh, I'll share. Okay, guys. So don't forget to fill out the evaluation survey, because if you don't fill this out, the academic affair will not issue your certificate for this power talk. It is very important. Okay, do not forget